Hello and welcome back to Samantha
and Gabby's
AP Bio channel. Previously, we talked about the light reactions. Now, we are here to talk about ATP and respiration. So, let's get started. Cellular respiration is how we derive energy from food, specifically glucose. And fun fact from the Amoeba sisters, Ranchos had one version, the chemical equation for cellular respiration is C6H12O6 plus 6O2, which turns into 6CO2, 6H2O, and energy, or glucose plus oxygen turns into carbon dioxide, water, and ATP. And our second fun fact, which will kick in later, fermentation is also the process that gives us drinking alcohol. Hashtag mocktails over cocktails, hashtag stay safe, and hashtag sobriety is sexy. And back to the regular program. First, our bodies need to turn energy into ATP by growing or moving around, etc. And we start off with ATP, or adenosine triphosphate. Adenosine is made up of ribose and 3 phosphate groups. And from the phosphate groups, once ATP takes off this phosphate group, it will then turn into ADP, or adenosine diphosphate. And from the breaking of bonds, it releases energy in the form of electrons. Then, due to water floating around in the cell, OH is what takes the place of the phosphate, which is the definition of hydrolysis. Hydrolysis is when water breaks down a compound. And now we shall turn it over to you.
Okay, so glycolysis is the breaking down of glucose under the presence of no oxygen, and it occurs in the cytoplasm. Glycolysis begins by the bonding of a glucose to an oxygen molecule, which then it is broken down and creates 2 3-carbon molecules called pyruvate. When the glucose molecule is broken, it releases electrons, and the electrons assist in the creation of ATP and NADH by bonding an ADP to a phosphate to create the ATP and the NAD plus to a hydrogen to create NADH. And because the glycolysis uses 2 ATPs and it creates 4 ATP molecules, the net total is 2 ATPs. And then under the presence of no oxygen, the pyruvates go to the fermentation, and the fermentation process needs more NADH. So over here, the NADH will be broken into NAD plus and a hydrogen, which is called NAD plus regulation. And when there is oxygen present, the pyruvate will be going through a transition intermediate step, which will transport the pyruvate into the mitochondria from the cytoplasm. The formation of ADP plus phosphate into ATP is called substrate level phosphorylation, which is a way for cells to quickly make ATP without oxygen.
To continue, the Krebs cycle happens across the inner membrane of the mitochondria, or the powerhouse of the cell, in the cristae. The cristae is the folds of the mitochondria's inner membrane that increases the surface area for the production of ATP. Then you will take a pyruvate, and you will create another 2 ATPs, which I'll put on the side, 2 ATPs per glucose molecule, and you'll form energy. 1 pyruvate is oxidized, and 1 carbon bonds with oxygen and leaves the cell as CO2. Then you're left with acetyl coenzyme A, or acetyl-CoA. And just to recap, enzymes are proteins that bring things together to react with each other. In this case, later on, we'll bring together a phosphate and ADP to make ATP for each pyruvate, and acetyl-CoA plus the 4 carbon molecule oxaloacetic acid, to make citric acid. To make citric acid a 6-carbon molecule, which will go here. But just to say what I left out accidentally, apologies. NAD plus comes along NAD plus comes along and combines with hydrogen to make NADH. Okay, to continue, citric acid is oxidized by cutting off carbons to get back to oxaloacetic acid. And at the end of the day, this is what makes the Krebs cycle a cycle. I don't know what I just said about the carbon cycle, please. Thank you. As carbons are cut off, it's in the form of carbon dioxide, aka your exhale. Each time a carbon comes off the citric acid, some energy is made in the form of NAD+, which is over here, and FAD. Imagine that's over there. NAD plus and FAD are enzymes that hold on to high energy electrons. Oh wait, Miss Lopez. Okay. High energy electrons and keep energy until they're later released in the electron transport chain. FAD and NAD plus then pick up hydrogens and energize electrons from each pyruvate, which creates a charge. Each pyruvate yields 3 NADHs. I'll make this 3 NADHs, 1 FADH2, and this is per cycle. And just to recap everything, glucose breaks down. I'll move this. Glucose breaks down into. Oh my lord. Okay, 2 pyruvates, 6 NADHs. I have to type 6 right there. 6 NADHs and 2 FADH2s.
Okay, and this brings us to the final step of cellular respiration. The electrons from the FADH and the FAD2s give energy needed to pump a channel of proteins across the inner membrane of the mitochondria. And this process is called chemiosmosis. And the pumping of proteins causes a proton gradient to be created on both the inner and the outer membrane of the mitochondria. Let's see. Proteins swap electrons to send hydrogens from the inside of the mitochondria to the outer compartment. Once out, protons want to come back to the other side of the inner membrane because of the protons on the outside, creating the concentration gradient. And then once the energy of proton flow drives mechanisms to squeeze some ADP and phosphate to make ATP, their process called oxidative phosphorylation. And then electrons from 10 NADH make 3 ATPs each, and 2 FADH2s make 2 ATPs each. And that's the end of cellular respiration.
Woo!